These are notes on the lactase lab that we're going to do in class on Wednesday or Thursday, depending on when you have class, because this is an early release week. So there's two compounds that we're going to talk about. One is lactose and one is lactase. So based on what you know about chemical compounds in biology, you should see that lactose ends in ose, so that's got to be a carbohydrate, a sugar. And lactase ends in ase, and so that's got to be an enzyme. And enzymes, of course, are proteins. So lactase is the enzyme that breaks down lactose. So lactose is a sugar that's composed of galactose and glucose. And both of these are simple sugar monomers. We studied them when we looked at the modeling kits, and so some of you may remember they actually have the exact same chemical formula, but how the atoms are bonded are different. So they act differently in our bodies, they have slightly different properties, they have a lot of similar properties as well. They're both carbohydrate monomers. But here we've got galactose, and here we've got glucose. And what lactase does, as I said, is it breaks the lactose. So this whole thing here is a lactose molecule. It's a polymer. And the lactase enzyme breaks the lactose into the galactose monomer and the glucose monomer. So people who lack the lactase enzyme don't have the ability to break down the lactose, and we would call those people lactose intolerant. And later this year we're going to look at in human history, actually lactose intolerance was prevalent. That's what everybody had. And then the lactose tolerance is actually a gene that has evolved pretty recently, which is kind of interesting to think about. And so we're going to talk a little more about that in a couple weeks. So enzymes convert what we call substrates into products. So we talked earlier this year about a chemical reaction, and we said the reactants to the products. If we're talking about something that's specifically converted by an enzyme, now remember, go back to your knowledge of enzymes. Enzymes don't actually change what's going on, they just speed it up. They catalyze what's going on. So the enzyme doesn't, it's not part of the reactants, it's not part of the products, it just helps the reaction along. And we call the things that are reacting the substrate when we're talking about enzymes, and then we call the end products the products, so that's still the same. So in the lactose example, the lactose would be the substrate, the lactase is the enzyme, and then the galactose and glucose are the products. And so you can see this here. Your substrate is the lactose, lactase is the enzyme, I'm going to circle that, and then the products are the glucose and the galactose. And so when they say the arrows represent the action of the enzyme, this is the enzyme splitting the lactose up into galactose and glucose. But remember that the lactase then is free to split up another lactose molecule. It's not used up, it can be reused again and again and again. It doesn't become part of the products or anything like that. And so here's a little animation for you. We've talked about proteins having specific shapes, and enzymes, of course, are proteins, and so you could see if this is the lactose molecule, it would fit very nicely here into what we call the active site of the enzyme, or the puzzle piece fits. So there it goes, and then it's broken up, glucose and galactose. Very fun. So this will help you understand the lab that we're about to do with lactase. It's going to be a lactase lab, and what we're looking at is how heat affects the ability of enzymes to do the work they need to do. So if you have any questions, put them on the YouTube comments or ask them in class. Have a good night.